last question. You have to write a lot, and the maximum mark for this is 6. What you have to do is to outline the ways in which liver regulates the chemical and cellular composition of the blood. Now the three important words. Liver, we have to look at this organ. The chemical composition of the blood and the cellular composition of the blood. How liver can regulate these two. Then we have to go back to our slide. Now, let's have a very quick reminder on the liver structure. Now, the yellow cells here represents the hepatocytes. Hepatocytes means liver cells. The yellow cells are the liver cells. And throughout the whole organ, you can see here, you can see a duct. There are a lot of ducts colored blue here. Now, they represent the hepatic sinusoids, or you can call it sinusoids. Now, in the sinusoid, there are some cells which is colored yellow in the diagram. Now, these cells are called Kupffer cells. They are there in the sinusoid to engulf bacteria and to break down all rubber cells. Okay, so bacteria and rubber cells, they are cells. That's why your liver is able to change the cellular composition of the blood. If they engulf bacteria, they will decrease the cellular number of bacteria in the blood. If they break down rubber cells, they also modify the cell numbers, the rubber cells number in the blood. Alright? Okay, now we have to look at another slide, this one. Now here we have an introduction on the functions of liver, including processing of nutrients. What about storage of nutrients in your body? The nutrients we're talking about here include the lipid-soluble nutrients, lipid-soluble vitamins like vitamin A and vitamin D. They can be stored in the liver if the amount is in excess. They're too much. Then the breakdown and recycling of rubber cells, just like what I told you before, by the Kupffer cells, the old rubber cells will be engulfed by the Kupffer cells and the hemoglobin will be broken down. Conversion of cholesterol to bile salts is also a task carried out by your liver. Don't forget, cholesterol can also be synthesized in our liver. And detoxification. Okay? Now, this one, the processing of nutrients, storage of nutrients, conversion of cholesterol to bile salts, detoxification, they all belong to the modification of chemical components in our body. While the breakdown and recycling of rubber cells, engulf of bacteria is the modification of cellular components in our body. So here, they all belong to the liver function. Alright, so now let's look at how we can put down our ideas point by point in the answers. I have grouped the marking schemes like this. These points, A and B, they describe glucose and glycogen conversion. So it is a chemical change, okay, carried out by the liver. Point C, talking about excessive iron can be, some nutrients in excess can be stored in the liver. So nutrient is chemical change, chemical composition. D, the liver detoxifies blood, so it is also chemical components. E, Kupffer cells engulf bacteria, so bacteria belongs to the cellular one, cellular composition of the body, of the blood. And this one, Kupffer cells break down rubber cells. Hemoglobin is split into heme and globin. Iron from heme is carried to the bone marrow to produce new hemoglobin in new rubber cells. Excess iron stored in the liver. Okay, now this is how the liver processes rubber cells, including both the cellular component and the chemical component. The next one, cholesterol, chemical, and plasma protein is synthesized by your liver cells, also chemical. Now this is the end of this paper. Thank you for watching.